So welcome to this talk by Anil Patri. Welcome for taking the long way from FDA over to Basel today. Anil is the chair of Nanotechnology Task and director of NanoCore at the National Center for Toxicological Research at US FDA, and we're looking forward very much to your talk. Thank you very much for accepting this invitation.
Thank you, Gerrit. You can hear me? Yes? Yeah, want to make sure. Again, good morning, good evening. It's a pleasure always to participate in ClinAm every year. This year we missed to be in person in Basel, but I appreciate Biat for organizing this meeting online so that we can update ourselves about what's happening in nanomedicine, nanotechnology area. As Gerrit said, my name is Anil Patri. I work for FDA. So the standard disclaimers apply. So the views expressed here are my own and not to be considered as official position or policy of FDA. So I'd like to update you all about a decade of progress in research, regulation, and policy at FDA. Often we don't get to showcase what we have done at FDA. And so we wrote an update to a progress report that was initiated in 2006 after the Nanotechnology Task Force was formed by the US FDA Commissioner. The task force itself is composed of members from all the centers and offices at FDA, and we meet every month. We also participate through the NNI, the National Nanotechnology Initiative, through interagency working groups within the US government. These are called the NSET and the NEHI subcommittee. And we have a lot of interagency collaborations because nanotechnology is a very broad field. And we cannot do it alone. So we do learn from experiences from elsewhere, from academia, industry, and also with other agencies. So when the task force was formed in 2006, the commissioner asked the task force to identify the knowledge and policy gaps at the time. This was almost 14 years ago. And to promote collaborative efforts in measurements, because nanotechnology at the time was kind of new for us at FDA with some products coming through, and we already approved many liposomes by then. But we needed to build that expertise and infrastructure within FDA and also collaborate with other agencies. And so this talk today is to summarize the progress that was made on the recommendations. And we released a public report in July this year on the state of nanomaterial science and technology, the research progress in infrastructure at FDA, and the guidances that we provided to the industry. We have 5 guidances that I'm briefly going to talk about. And we spend around 10 to 12 million every year, internal research grants, and also staff training and professional development is critical for us because as new technologies come through that utilize nanotechnology, the reviewers need to be appropriately trained, and we do that every year. And also domestic and I'll cover some domestic and international coordination and standards development activities at FDA. You can download this report. I have the link at the bottom of the page and at the Nanotechnology Task Force website at FDA. So we do conduct a lot of research at FDA. We call it regulatory science research, slightly different from basic fundamental research, and mainly on FDA questions, FDA problems that we come up with through the gap analysis and internal review of submissions and see where we need to put our resources, and also horizon scanning, attending meetings and looking at what is coming through. And this research then leads to the guidance documents, because expertise is required. So we gain that knowledge through internal research and also staff training, as I mentioned, and standards development activities. We also have standards recognition program that recognize the standards from elsewhere, from standards development organizations. So this really resulted in a huge impact for us at FDA. FDA scientists are co-authors on more than 500 peer-reviewed publications. We have more than 120, actually close to 150 staff trained in nanotechnology in different centers. And also we reviewed more than 800 submissions, and they are mostly drug products, but then there are many devices and other submissions. As I mentioned, 5 final guidances and 1 draft guidance are issued, and we are very heavily involved in standards review as well as standards development, meaning if the standards are developed at ISO, OECD, ASTM, those standards come through for review, and we have experts, subject matter experts that review and comment on those. And we did recognize 16 nanotechnology standards, meaning companies can come through submissions using these recognized standards, and so the communication becomes that much easier. This is through the Center for Devices, but also Center for Drugs, we call it CEDAR, also is setting up a standards recognition program. So all these years, almost 10, 15 years, this work really had a huge impact in our work and to prepare ourselves for current and future submissions of nanotechnology-based products, drug products, devices, and others. Many of you may have seen this if you listen to talks from FDA. So we have seen a gradual increase in drug product submissions. The last update was a few years ago, the publication in Nature Nanotechnology, and that was in 2017. And so that data had publications still, or the submissions till 2015, 16 or so. So since then also we have seen an increase, and if this is the latest data until 2019, as you can see, the green bars, which are generic drug product submissions, they're gradually increasing. As you approve the products, many of them become generics, and now we have seen uptick in the green bar, the ANDAs, more than 700 product applications, and more than 70 NDAs and ANDAs were approved. So 70 products containing drug products containing nanotechnology were approved. The blue bars here stand for INDs. These are the investigational new drug applications, meaning these are, some of them are obviously in clinical trials, and then they eventually get approved. The red bars are the NDAs. And so we have seen a gradual increase in the complexity of these drug products with the multifunctional, multi-component structures and devices. And our policy allows us to be flexible and to approve innovative and promoting innovative nanotechnology and promoting beneficial nanotechnology product development. So within these products, you can see most of them, or 1/3 of the submissions, are liposomes, but we also see nanocrystals, emulsions, iron polymer complex. Gerrit actually did a lot of work on those generic drug products as well, and he leads that work in Europe. And then many polymer-based products as well. Cancer is still predominantly a major indication, 1/3 of those submissions based on cancer, but also inflammation, infection, and some imaging agents. The route of administration of these in drug products is predominantly intravenous, more than 60% to be intravenous, but we have seen other modes of drug products, including oral, some ophthalmic products and emulsions, inhalation products. So overall, we have seen a wide variety of submissions, a wide variety of compositions in the drug products, and these are very complex to develop in many ways. Complexity increases with increasing the more recent of the products that are becoming more complex with these multi-component systems. Apart from the drug products, we have seen many devices, and here are some examples of those devices. As you can see, many of them utilize metals and metal oxides, such as silver, gold, silver for mostly antimicrobial, antibacterial coatings on catheters, wound dressings, and we have seen others in dental fillings and titanium oxide used in many devices, nanostructures, and dental fillers that use silica nanoparticles. Just to give a flavor of the kind of submissions that we have seen in the devices space. And these devices are also increasing, a gradual increase in the submissions, whether they are 510K submissions or pre-market approval and others in the devices space. But also we have many veterinary drug products, food ingredients, the cosmetics, sunscreens, and vaccines. And so FDA knowledge has increased with time in various areas, not just the drug products, but you can see the variety of submissions because FDA has a broad mandate of regulatory work in US. Coming back to the drug products, as I mentioned, they involve complex physicochemical testing. So many of our resources within FDA are on this complex CMC or the chemistry manufacture control section of an IND filing, which is physicochemical characterization and testing. We know that slight change in these parameters may have significant influence in the biological properties, whether they are immunotoxicology, a general toxicity, safety, or efficacy. So appropriate validated methods are to be used for these testing. And most importantly, that these methods should be able to discriminate between acceptable and unacceptable batches. And we call them the critical quality attributes. So most people talk about size polydispersity, but this is only one factor, as most of you that develop the drug products know. There are multiple techniques that are recommended, even though dynamic light scattering is most predominantly a common technique that is used, and it has its advantages, but also has limitations for polydispersed material. So define the product using multiple techniques, could be electron microscopy or separation techniques, such as asymmetric flow, field flow fractionation, or size exclusion chromatography, and so on. Or laser diffraction, for that matter. Sometimes they go beyond the size, and so those agglomerates become, the characterization of larger agglomerates become a factor to be considered. Apart from the size, the surface characteristics are also very important because they influence binding the biodistribution in vivo. Zeta potential is a common way of looking at that electrophoretic mobility, but it has its limitations. You cannot measure the zeta potential at higher ionic strength or more complex buffers. But also if the particles are coated, one could look at either thermogrammetric analysis or LC techniques to look at the quantitation, or quartz crystal microbalance, for example, to look at the binding of these ligands. As they contain drug products, the free or unencapsulated drug products versus encapsulated drug products is a big question, because the pharmacokinetics of the free drug is going to be different from the one that is encapsulated within the nanomaterial. And that definition, and there are methods, standardized methods now that one can differentiate between, let's say, protein-bound drug, free drug, and nanomaterial encapsulated drug. And there are methods also to release testing for these drug products in vitro. The other factors include excipient testing. Sometimes the synthesis has some impurities, and just like any other traditional drug products, those impurities testing is also an important attribute, as well as stability. Are they stable for 1 year, 2 years? Some of these emulsions can phase separate, and so those release testing is part of the submission process at FDA. So within FDA, we have excellent research infrastructure. We have at both the White Oak campus, which is outside Washington, DC, as well as in Jefferson campus, where the National Center for Toxicological Research is located. And those facilities are collaborative centers or facilities between multiple centers so that we could do collaborative work within FDA. And they have advanced equipment instrumentation in electron microscopy, optical microscopy, spectroscopy, chromatography, all kinds of scattering and diffraction techniques, and so on. So all these infrastructure facilities would provide us with opportunities for extensive collaborations, both within FDA and also outside FDA. And these are usually funded through the center funding. As you can imagine, the amount of money it takes to maintain those facilities and scientists. But also we give internal grants within FDA. These are not external. FDA is traditionally not a funding agency, unlike NSF and NIH. But we have internal granting process, and these are called the cores, or collaborative projects. And we gave so far, as of last year, more than 40 grants. And as you can see, they average less than, let's say, $1 million, maybe 800,000 the last few years. And the total funding that we spent on this is around $6 million. Every year we spend between $10 to $12 million on total research at FDA and other infrastructure support. So these projects that we have done internally include, for example, a detection, identification, and characterization of nanomaterial in FDA-regulated products. Many of the drug products have pre-market approval, meaning one has to come with a submission, let's say an IND submission that we get to see. But then there are many, let's say, food containing products in food, for example, or sunscreens and other, where we need to characterize material or detect nanomaterial in some of these products. And so we develop methods, and again, identify critical quality attributes and how structure of these material influence biological behavior. Because once we understand that, then we can ask appropriate questions to the sponsors of these studies. We also do methods and methodology development, which eventually end up with the standards through international bodies. And also we conduct in vitro studies, in vivo studies, many kind of toxicity study, including genotoxicity, effects on microbiome, pharmacokinetics, and some efficacy studies in appropriate animal models. Staff training is, again, an important aspect within FDA, that we need to train our staff as the new technologies come through, new kind of submissions come through. So we have an annual training at FDA. This is called the hands-on nanotechnology training for reviewers and scientists. And this is done collaboratively between multiple centers, between NCTR, ORA, CDRH, and CDER. These are multiple product centers as well as research centers. They contain lectures and lab sessions, meaning they actually get to see the equipment, they actually operate the instruments to actually see what kind of data that comes out, so that reviewers can understand how to analyze the data sets and ask appropriate questions, and both covering the basics and as well as more advanced lectures from lessons learned and pitfalls elsewhere. And as I mentioned, 150 folks went through this training, but also we have many postdocs. These are called ORISE fellowships that we give, and also lecture series with subject matter experts. We don't always know everything. Obviously, we don't have the capabilities to understand everything at any given time, but we have the knowledge and experience within FDA with the past product submissions and approvals. And so we do bring academics to give seminars so that we can understand what is coming, what is new, what is cutting edge. And also we have many public workshops over the years, starting from 2006 or so, multiple public workshops to get input. And also we had sabbatical and internships, both from students as well as scientists from other regulatory and research agencies across the globe. So this really helped us retain highly knowledgeable staff within FDA, and as we continue with the new products review as they come through for multiple centers. Coming to the guidances, we have 5 guidances, and I'll go through them, but the main guidance is an overarching guidance considering whether an FDA regulated product involves application of nanotechnology. Early on, there was a discussion as to what that size should be, and most of these discussions stop at size. And as you know, if you have a drug product, it is a lot more than size and the drug that one is used and surfaces and other factors. So based on the submissions and based on the regulatory authorities, we wanted to keep it flexible so that if someone is developing a product that is, let's say, beyond the 1 to 100 nanometer size range, then the regulation is still flexible. So we did not establish a regulatory definition for nanotechnology, and some broad screening tools are used for FDA purposes that recognize FDA's interest in properties extend, as I mentioned, beyond size. And this provides for tailored approaches that adhere to applicable legal framework and reflect the characteristics of specific products and product classes that and evolving technology and scientific understanding. So this makes it very fluid, very flexible, both for industry that is coming through submissions as well as for FDA. But we do have 2 points to consider. As I mentioned, there is no regulatory definition, but then we do identify, or we do understand that the nanotechnology or nanomaterial are different, in the size being one of the main parameters. So we consider 1 to 100 nanometers if the material or end product is engineered, the keyword is engineered, or designed to have at least one dimension within that size range, approximately. And also beyond that size range up to 1,000 nanometers or 1 micron, where the product is engineered to exhibit properties, phenomena that are attributable to the dimensions, even though they fall outside that 100 nanometers range. So this flexibility really helped us as well as industry. So we have seen many submissions beyond the 100 nanometer size range where there are unique phenomena, unique characteristics that come through. And we have seen products with 200, 300 nanometer size range. And these guidances include the overarching guidance, and then the guidance on safety of nanomaterial in cosmetics, and then the guidance on significant manufacturing process change of emerging technologies in food and food contact substances. And also we have a final guidance on food for animals that use nanomaterials. And there are many guidances in liposome drug products, in chemistry manufacturer control section, pharmacokinetics and bioavailability, and then product-specific guidances. There are, for example, many liposomal drug products. And so apart from the overarching guidance on liposomes, we also have specific guidances on different products. So the latest guidance, which is still in the draft stages, meaning it'll be become final, it is publicly available. We call it CDER guidance that is produced from Center for Drugs and Center for Devices on drug products, including biological products that contain nanomaterial. So we have, beyond these, we have many other guidances. For example, guidances in polymers, guidances related to devices, and that can be used, not specific to nanotechnology, but they are applicable. Again, they can be downloaded from the task force or FDA website. We also early on decided that standards are going to be critical. And so we got involved in standards development activities. And more recently, we formed a standards subcommittee within the nanotechnology task force in 2016 with experts, and came up with a prioritized list of standards in the global summit in 2016. And so we have been methodically working with the stakeholders in developing those standards that are identified by regulators from across the globe and industry as well, contributing to the standards development. The impact is that by actively participating in standards, we can collaboratively develop these through the stakeholders. We reviewed and commented on more than 55 nanotechnology standards and recognized 16 standards. And also Center for Drugs is initiating a new standards recognition program. So we are actively participating in ASTM E56, ISO TC229, and OECD Working Party on Manufactured Nanomaterial committees. And within FDA, we are very active in actually doing the lab work. We do interlab studies and with stakeholders, and the two standards here at the bottom, the cryo-TEM of liposomes, because liposomes are a huge class of compounds at FDA, and also the chemotaxis assay, one of the in vitro assays, became standards through the work of FDA staff. There are many work items that are going through this development process that we are involved in. Apart from this, we have interagency coordination through the NNCO, NNI. We have the NCI NIST FDA MOU in the nanotechnology space. And this was established early in 2004, 16 years ago, but renewed every 5 years. The latest is renewed 5 years ago, in January this year. We have collaboration with the National Toxicology Program, and some of the standards work is funded by them. Consumer Product Safety Commission, or CPSC. We have overlapping space in food contact material. So we have collaborations there. More recently, we have an interest in nanoplastics, and we are part of this nanoplastics interest group, which is composed of more than 20 federal agencies, and coordinate and collaborate as required to address the global issues related to nanoplastic. So as you know, nanoplastics are formed through degradation of bulk plastics, and they eventually end up in the food stream, whether seafood and food material. And so we have an interest in that space, and we are working with others, and also organize a global summit. The last year global summit in Italy, GSRS 19, was on nanotechnology and nanoplastics. But we advance this regulatory science research globally through this GCRSR. We have global summits every year on different topics, and every 3 years through this global coalition the topic is nanotechnology. Starting in 2013 and 2016, we focused on standards, and last year on nanotechnology, nanoplastics. So through these, and as you can see from the graph on the left side, all these regulatory agencies from Europe, Asia, North America, and South America are part of this global coalition. And so we do discuss, we inform each other as to what is coming so that we can better prepare ourselves. And these international interactions are also through the IPRP, Nano Working Group, International Pharmaceuticals Regulators Program. And we usually meet every year at CLINOM in Basel. And many of you may have seen us presenting the IPRP working group. And today, actually, we are meeting later this week online. We also have specific interactions with other regulators such as Health Canada, European Food Safety Authority, Canadian Food Inspection Agency, and so on. And we have these capacity building and workshop activities that train each other. So we learn from their experience and they learn from our experience. Specifically one interaction I would like to highlight is an Indo-US nanotechnology workshop. For a couple of years, we had these collaboration in workshops with Indian regulators, the CDSCO, or the Central Drug Standards Control Organization, that ended up with India developing guidelines for evaluation of nanopharmaceuticals. This was released last year in 2019. So these global interactions are critical for FDA because products that we regulate come from all over, not just from within US. There are many emerging,
Can we wrap up in a minute, please?
Yeah.
We have to.
Yes. So there are many emerging products that are very complex, multi-component products with the convergence of emerging technologies. You also have seen many gene delivery systems, vaccines that are coming through that utilize nanotechnology. And so we are prepared within FDA to regulate or have a dialogue with industry to review these emerging products. So in the future, we continue with these activities I described here, including research standards, and then continue with the international collaborations as well as domestic collaborations. So I'll stop here with an acknowledgement of FDA offices across the globe, Europe office, India office, and my nanotechnology task force and researchers within FDA. Thank you.
Thank you very much, Anil. Very nice talk, very enlightening. Can I start first with a comment? Because you said I was working on generics. Actually, we compared different products that were supposed to be generic, but we found they were not generic at all. So I try to come to terms with the FDA's term of complex generics. I'm still not a big fan of it. I like the term nanosimilars more, but that's a long discussion we could have here. Let me take a question from the audience, please. It's from Jean Fontaine. You already talked a lot about the interaction with other organizations and other regulatory authorities and so forth. But the question is, you probably could be more specific. The question is, do you connect these standards with other standards, such as the ISO identification medicinal products, which are then used in regulatory information exchange, and how are then they interrelated? Very.
Yeah. So the one that thank you. That's a good question. So the one that I talked about, the ISO TC229 or ISO TC24, these are kind of specific to nanotechnology, and we work in that space. But obviously, those are not limited to those. For example, the United States Pharmacopoeia, the European Pharmacopoeia. I mean, there are many other standards within the, for example, ISO or ASTM we work with. And so products coming through submissions utilizing those standards and those that are recognized by FDA. So the standards recognition program is really the one that that helps the industry, but that doesn't mean that industry cannot come with others if they validate their methods. It does not limit industry. So yes, we do work with other standards, other regulatory agencies. FDA has more than 700 scientists in many regulatory bodies. I mean, many standards development bodies. So we utilize all those standards.
Right. Okay, thank you. I have another question. You mentioned that in 2003, nanotechnology was still somewhat new to FDA, even though even before 2000, there were already products or submissions been submitted. Now we are in a phase where you alluded to that, where everybody and their mother are developing delivery systems for DNA, RNA. We have a Nobel for CRISPR-Cas. How far are you prepared if I were to try to put on the market or give a submission for a DNA vaccine against COVID-19? How much are you prepared to accompany this?
Yeah, so that's a good question. So you're right. So we approved, or FDA approved the liposomal drug product, let's say liposomal doxorubicin in 1995. There are many polymer drug products that were approved before that. Before we used the term nano. If you search the term nano, before 1995, you may not come across any publications, but that doesn't mean that we don't have that expertise and experience. But what I meant is that the new kind of nanotechnology, new kind of nanomaterial, that, for example, a gold nanoparticle with something on the surface, this is something new to FDA. So how do you then ask questions about polydispersity? How do you ask questions about the CMC or the core shell nanoparticles? Or, these are quite complex particles. What I mean by not knowing is that even though we have traditional knowledge and experience for a long time and internal expertise, these are some of the new ones. For example, electron microscopy is not used in other submissions, drug product submissions, but then that becomes a new way of looking at things. So we certainly is evolutionary in the sense that we have in-house expertise. We do learn from prior knowledge and experience that we have internally, because when they go through clinical trials, we have a lot of interaction with the industry, and we learn a lot, and all that knowledge is kept inside. And so we are prepared. For example, you mentioned about COVID vaccines, and some of them utilize lipid nanoparticles, as many of you know, RNA, mRNA. So we are highly prepared, and then we obviously got them approval through the clinical trials now. And so any new technology is okay in the sense that we have existing laws and regulations as well as internal expertise. But we learn and work with the submitters. That's why if it is new, we suggest early consultation so that we can have the dialogue with the sponsors and help take the product through the process, preclinical to clinical process.
Okay, fair enough. As you know, complex systems are my interest, also my academic interest, and we start to fool around a little bit with very complex stuff with, I don't know, antibodies connected to liposomes. Will we ever have a system like that on the market, like a targeted liposome targeted by, I don't know, by antibody or antibody fragments?
Absolutely. So we already see, we have already seen, let's say, targeted drug delivery system come through. That's what I meant by the evolution of these products from more simpler drug delivery systems to more complex systems where you have one or other kind of targeting ligand and one or more drug products or drugs in the system with a nanomaterial. And so absolutely, and that's our most advantageous, and that's one area that I think that a more resource can be put in. Because most of the products you see are simpler systems. Of course, even within those simpler systems, you increase the safety profile in many cases. Even I was watching some of the lectures yesterday in Clinom. Many of these products are formulations in a way that there is some toxic issue, and then you reformulate, reduce the toxicity. But the real advantage of nano is that utilizing the size surfaces, targeting and multifunctional capability with multiple drugs encapsulated to have synergy for new disease or the diseases that don't have any drugs, and that area is really ripe, and so we are prepared, and we are looking for those submissions to come through.
Very good. I think if there are no other questions from the audience, Anil, let me just say I was very sad that I couldn't meet you in person today and everybody else at Cleanon, and I hope that we can have this experience next year. All the best for you, and hope to see you soon in person or by Zoom. Thank you very much.
Thank you.
So to all the audience and of course the organizers, thank you.
Thank you.